The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and welcome to Lab Manager's Product Showcase webinar series. I'm Tanuja Kopal, contributing editor for Lab Manager, and I will be moderating today's webinar, where we will be discussing the use of mass spectrometry for qualitative and quantitative analysis. The webinar today is sponsored by Agilent Technologies, and we will be joined by two technical experts from the company who will be talking about recent innovations in mass spec instrumentation and how you can further increase the throughput and efficiency of mass spectrometers in your lab. Before we begin the presentations, I want to remind you that this webinar is meant to be very interactive, and we would like you to take this opportunity to ask questions and have them answered by our experts in real time. You can send along your comments or questions anytime during the webinar by typing it into the question box that appears on the right-hand side of your screen. We will begin addressing your questions after our speakers have given their talks. Today's webinar will also be available for viewing on Lab Manager's website, which is www.labmanager.com, for at least 6 months. The website also hosts a number of resources, such as technical bulletins, application notes, and white papers that you may find useful. In our closing slide, we will also share with you the email addresses of our speakers, so you can reach out to them for a copy of their slides or for any guidance on this topic. So with that, let me turn it over to our first speaker, Dr. Yuqin, who is a senior application scientist in the division of LC-MS marketing at Agilent Technologies. Her current responsibilities include planning marketing strategies for LC-MS applications in the small molecule pharma segment and leading the design and delivery of integrated LC-MS application solutions with global teams. Her talk today will focus on the use of Q-TOF LC-MS for metabolite identification. Welcome, Dr.
Dai. Thank you, Tanisha, for the introduction and arrangement of this webinar. The topic I'm going to discuss today is the Quant workflow by using 6550 and final Q-TOF LC-MS for simultaneous determination of metabolic stability, metabolite identification, and profiling of 1 micromolar Buspirone in rat liver microsomes. Pharmaceutical industries are continuously looking for the increasing productivity and reducing cost. So how can we use our limited resource to achieve this goal? One of the solutions we believe it can help you to achieve that goal is the Qual and Quan solutions. So it combines the qualitative analysis and the quantitative analysis in one single experiment so that you can obtain more information with less work involved. Now the question is Quan on a single analysis feasible? What is the instrument requirement? So in order to accommodate qualitative analysis and quantitative analysis in a single experiment without loss of data quality, mass spectrometer has to offer the high sensitivity equivalent to the triple-quadruple like performance for the quantitation. Secondly, mass spectrometer has to also offer the scan speed fast enough to accommodate the narrow LC peak width to achieve the rigorous quantitation, as well as to meet the expectation needs for the throughput. Furthermore, the accurate mass and high resolution are required in order to achieve the reliable identification and high selectivity for the quantitation. Last but not the least, the dynamic range needs to be broad enough, typically requires 3 to 4 odds in order to meet the expectations for the most applications. So Agilent, with technology innovation, Agilent has developed a new 6550 final Q-TOF mass spectrometer. This new platform incorporates a number of new technologies, this including the Inferno technology, which provides the 10-fold increase in the sensitivity compared to our previous model 6540 Q-TOF. the dual Agilent robust and stable mass calibration through the entire runs. The ion beam compression and shaping provides the greatest sensitivity while maintaining the high resolution of up to 40K and sub 1 ppm mass accuracy. The new electronics and the software algorithm enables the faster data acquisition rate of up to 50 spectra per second. So these new technology innovations result in the dramatic increase in the sensitivity. This example shows the result for our benchmark compound, Reserpine. As you can see, the extremely low detection level at 25 femtogram on column was achieved with signal to noise ratio of 49 and resolution of 31K and the mass error of 0.1 ppm. 6550 and final Q-TOF also demonstrates the excellent quantitation performance for the biological samples. What you see here is the calibration curve of the Buspirone in red plasma. Over 3 odds of linear dynamic range was achieved with low limit of quantitation at 12.5 picogram per milliliter. The linearity is excellent. The R-square is 0.998. Furthermore, the assay accuracy and precision for all the calibration stands and low and high QC are well within the bioanalytical acceptance criteria. So overall, 6550 and final Q-TOF can offer high performance needs for both qualitative and quantitative analysis. Here is a brief summary of the key features. The sensitivity is at a low picogram per milliliter level. The mass accuracy is excellent with mass error less than 1 ppm in MS mode and less than 2 ppm in the MS mode. Resolution is up to 40K and the scan speed is up to 50 spectra per second in MS mode and 30 spectra per second in MS mode. While the linear dynamic range is also achieved, typically in the range of 3 to 4 odds, with the in-spectral dynamic range of up to 5 odds. So next, I'm going to discuss a case study to demonstrate the utility of 6550 and final Q-TOF for simultaneous determination of metabolic stability, metabolite identification, and profiling. This workflow shows a classic approach for metabolic stability, ID, and profiling assays. Traditionally, the quantitation such as metabolic stability and metabolite profiling is often performed on the LC triple-quadruple mass spectrometer using a physiological relevant substrate concentration in the range of 0.5 to 1 micromolar. For the qualitative analysis, in this case metabolite identification, a different LC tandem mass spectrometer was often used because this is a non-targeted analysis. So due to the sensitivity limitation of the traditional LC-MS, relative high substrate concentration in the range of 10 to 20 micromolar is often required in order to identify metabolites with a broad coverage. So with the great performance enhancement, 6550 and final Q-TOF enables us to combine these 3 assays in one experiment with one substrate concentration at 1 micromolar for both quantitation and identification. This diagram shows how high-resolution accurate mass Q-TOF data can be utilized for the quantitation as well as for the qualitative analysis. Basically, we leverage the high mass capability of the Q-TOF, and we use for the quantitation, we leverage the high mass capability of the Q-TOF we use a very narrow mass extraction window, typically in the 10 ppm, to obtain high selective MS data for the quantitation. What you see here, the metabolic stability profiling and the metabolite profiling data were obtained from the accurate mass Q-TOF data, MS data. For the qualitative analysis, in this case, metabolite identification, so we identify the metabolites and determine the modification sites based on the accurate mass Q-TOF MS data as well as the MS data. So the Qual workflow starts with the single injection of biological sample to the LC-MS, and the data acquisition includes full MS scan followed by 2 to 3 MS scans. Once data acquisition is completed, data are processed by using a package of softwares. This includes Metabolite ID software, molecular structure correlator. These two softwares are used for the identification and the structure elucidation. MassHunter quantitative analysis software is also used for quantitation. So finally, the metabolic stability profile and the metabolite profiles can be established based on the quantitation results. And the Metabolite ID report and the Quan report can be generated accordingly. So here is a brief summary of experimental conditions. The 1290 Infinity UHPLC coupled to the 6550 and final Q-TOF was used for data acquisition. The mass spectrometer was operated in positive ion mode with full MS scan followed by 3 auto MS scan in this case. The scan rate is 10 spectra per second in both MS mode as well as the MS mode. This was optimized to achieve the rigorous quantitation as well as maximum sensitivity. The LC flow is 0.4 milliliter per minute with a very generic gradient with runtime of about 3.5 minutes. The Agilent C18 column was used for the separation. The Buspirone concentration is 1 micromolar, and the Buspirone was incubated in the rat level microzones at 37 degrees for periods of time. The reaction was stopped by adding the acetonitrile at 1:2 ratio. So high mass accuracy is the key to the reliable identification. What you see here is the results from a 10-minute incubation sample. The top panel is the MS spectrum for one of the monohydroxy metabolites. The bottom panel is the MS spectrum for that precursors. So we not only achieve the excellent mass accuracy for the precursor ion, in this case mass error is 0.11 ppm, and the molecular formula was generated based on the accurate mass. We also achieved the excellent mass accuracy for all the fragment ions. In this case, less than 1 ppm mass error was achieved for most of the fragment ion. Furthermore, the high isotope fidelity was achieved with match scores greater than 97% for mass, isotope abundance, isotope spacing match. So this triple match further gives the high confidence for the metabolite identification. This example demonstrates the compatible results between the triple, quadruple, and the Q-TOF. As you can see, both sets of data correlate very well, even at levels where parent less than 0.1% of parent remains. So to ensure the Quan workflow highly efficient, a good software package is equally important. So in next few slides, I will briefly discuss how MassHunter software can facilitate the data processing for both quantitation and metabolite identification. This flow chart outlines the Metabolic ID workflow. Basically, the MassHunter Metabolic ID software starts with an untargeted approach by using molecular feature extraction algorithm, in short MFE. So by using the molecular feature extraction algorithm, it finds all the compounds in sample as well as in the control, and then the software compares, this algorithm compares both sets of data and then find the differences and then give a list of potential metabolites. Those potential metabolites are further confirmed or identified by multiple algorithms. This includes the mass effect filter algorithm, find the compound by auto MS, and molecular formula generation, and isotope match, and so on. The structure elucidation is facilitated by using the molecular structure correlate software and the Novacia auto-shift algorithm. I will discuss these two tools in next few slides. Finally, the identified metabolites are flagged, and the user can review and conclude the metabolites based on the Metabolic ID results and your knowledge. So this is a compound-centric data navigation window, and user can set up a method through this pull-down menu and in a logical sequence, and then click Find the Metabolites. So software will process data and finally give Metabolic ID results showing in this window. This column lists a number of potential metabolites based on the molecular feature extraction comparison, and the results showing here are obtained from the individual algorithms. So if those potential metabolites are identified or confirmed by those individual algorithms, and the green box and the red or blue checkbox will be displayed. So if potential metabolites are not confirmed or identified by each individual algorithm, then the red color or yellow color will be displayed. For example, this column, this algorithm monitors the UV signal. In this experiment, we don't have UV detector connected. That's why I explain why negative results are showing up here. By click each individual metabolite, and this window will display the chromatograms or the spectra by click this pull down menu to select the option. So what you see here is an overlaid MS spectra of the parent molecule and the one of the metabolites. Novacia auto-shift algorithm will indicate which fragment ions have been shifted. So in this case, these two fragment ions have been shifted. And it will also indicate how many mass units have been shifted, and also indicate the possible biotransformation. So in this example, actually, possible biotransformation is oxidation. This table will list all the MLZ value of the fragment ions and the mass shift. In this case, the mass shift is 16 mass unit. So one of the important I would like to point here is our MassHunter Metabolic ID software provides a very comprehensive detection of the expected and unexpected metabolites through a wide range of the algorithms. So the benefit of using multiple algorithms is it can prevent the exclusion of the potential metabolites. For example, this ND-alkylated metabolites was not identified by this mass defect algorithm. So user can further look into it and find out whether this is a false result or due to some other reason. In this case, what I found is the initial mass defect window I set up is 40 milliDalton. So for ND-alkylated metabolites, typically mass defect window number value are greater than the 100 milliDalton. So this explains why this algorithm shows negative result. This really provides the high confidence in metabolite identification. So another important software tool I would like to briefly discuss here is molecular structure correlator, in short MSC. This is a very useful tool to facilitate the structure assignment for the fragment ions. The unique part of this software tool is it predicts the structure based on the experimental MS data. Therefore, the results are more relevant and make sense. What the user needs to do is upload the experimental MS data to the software. And if you know the structure, you can input the structure. If you don't know the structure, you can do database searching, for example, ChemSpider. So in this particular case, because we study Buspirone drug metabolism, so we know the parent molecular structure, and then the next thing you need to do is just click MSC, and then the software will calculate the molecular formula generation score based on the accurate mass of each MS fragment ion and its neutral loss from the precursor ion. So it will display all different formulas based on all this information. Typically, the high scores is the winner. So software will also calculate all the possible subformulas for each fragment ions with mass deviation in ppm. For example, in this case, M/Z at 122, this fragment ion shows 3 subformulas. And the top one shows the lowest mass error at 0.4 ppm. So this is the most relevant, and therefore this formula was selected. The software will also predict the possible structures for each fragment ions based on the accurate mass and the penalty score, meaning how easily the bond can be cleaved. So in this case, there are 4 possible structures for this fragment ions, but this one shows the lowest penalty score, and will be and is selected. Of course, when you make final judgment, you also need to, based on your chemistry knowledge, and all this prediction. So by using that approach, all the substructures are assigned to each fragment ions of the Buspirone. So now we determine the substructure for the fragment ions for the parent molecule, and we know how many mass shifts based on the Novaci auto shift algorithm. Therefore, we can easily determine the modification size of a metabolite. In this case, this most likely happen on this carbon-carbon bond. So once metabolites are identified, we can easily extract those ions from the total ion chromatograms because the Q-TOF full MS scan provides the full spectrum through the entire run. What you see here is a representative overlaid ion chromatograms from a 10-minute incubation sample. And you can see the major metabolites identified are oxidation metabolites, a number of ND-alkylated metabolites were also identified from this sample. So after identifying all the metabolites, next step is to do quantitation for all those metabolites. This is a mass setup window of the MassHunter quantitative software. The mass setup actually is pretty simple and quick. In this case, what the user needs to import is the M/Z of the parent molecule and all the metabolites, and also need to input the retention time and the mass extraction window. And then software will process data in automatic fashion in the batch mode for all the multiple time points of the incubation samples. Finally, the metabolic stability profiling and the metabolite profiles were established based on the Qual results. So in a summary, 6550 and final Q-TOF is well suited to the Qual Quan workflows, excellent sensitivity is demonstrated with detection of buspirone and metabolites at all time points in the 1 micromolar buspirone incubations. This workflow also meets the throughput expectation with runtime of about 3.5 minutes per sample. The high mass accuracy offers high selectivity for confident identification and reliable quantitation. By combining the qualitative and quantitative analysis in a single experiment, it offers more information with less work involved. Therefore, this workflow has the potential to increase the assay productivity and reduce the costs. So I will end here, and thank you for your attention.
Thank you, Yuqin. If you have questions based on Yuqin's talk, please send it along. And you can do that by typing your questions into the question box that appears on your screen. Our next speaker is Dr. Jennifer Rossi, who is the RapidFire Program Manager at Agilent Technologies. She has spent her career in strategic marketing at life science companies both in the US and the UK. And prior to her role at Agilent, Jennifer was the Vice President of Marketing at Bioceus Life Sciences, which is now a part of Agilent. Welcome, Jennifer.
Thank you very much, and thank you everyone for joining. So you can just talk to everyone a little bit about using Qual Quan for more efficient and better mass spectrometry analysis. So what I'm going to talk to you about is increasing the efficiency of your mass spectrometer's throughput, and doing this using something called the RapidFire high-throughput mass spectrometry system. So I'll talk a little bit about an introduction to the technology. Then I'll tell you a little bit about some of the applications for which RapidFire is heavily used. Lead discovery, in vitro ADME, two of our main applications, and then talk a little bit about some demonstration data on those applications, and then finish up with some brief conclusions. So this slide is meant to show you that RapidFire has been in the industry and available for several years. Our first instrument was placed in 2007. However, there's been an evolution. So the RapidFire instrument was first part of the company Biotrove. This company spun out in 2009 to form Bioceus Life Sciences. And then almost a year ago, so on March 1st, Bioceus and the RapidFire technology was acquired by Agilent Technology. So RapidFire is now a part of Agilent, but if you had heard it as a Biotrope or Bioceus product, just to assure everyone, it's still the same product and still the same group of people, just now we're all under Agilent Technologies. And we've always been committed to enabling faster and more efficient mass spec analysis. So a lot of this history is down here. And very importantly, we're used by all of the top 10 global pharmaceutical companies. So RapidFire is a very prevalent instrument in the industry for drug discovery. Now I want to talk a little bit about how RapidFire is able to take additional advantage of your mass spectrometer for more efficiency. So what is it that's so great about mass spectrometry? Well, it's very sensitive and selective. One of its main advantages over traditional assay methods is the fact that mass spec is a true label-free detection, where the compounds of interest are directly measured in a quantitative manner. So that's kind of what we're looking at here on this slide. So there really is a value to a native reaction substrate and product measurement. So for example, this diagram here, you can see mass spec is detecting this phosphorylated compound of interest, where if we were using an optical assay, this would rely on a tagged molecule, proximity to another molecule, and release of a fluorophore. Such a complex set of interactions can lend itself to things like background, false positives, that may confound some of your data. But so the mass spectrometer then enables this direct quantification. However, there are, of course, some limitations to mass spec. In order to detect a molecule in the instrument, the molecule must be charged. There requires a desalting step to remove the assay milieu of some buffers that the mass spec that are not compatible with the mass spec. And this purification is generally achieved by chromatography, often liquid chromatography, as you just heard Yuqin talk a lot about, which can be somewhat time-consuming, especially if you're comparing it to an optical assay per se, where we're looking at a plate reader, and these plate readers tend to read very quickly. The other limitation of mass spec is that it's fairly expensive. This is a big piece of capital equipment, and the scaling this workflow can be somewhat complex if you need to continue to meet demand in your laboratory by adding additional liquid chromatography mass spectrometry systems. So what the questions we're going to try to answer here, or that many people have been trying to answer, I should say, is that how we can maximize this mass spec resource, or how can we make liquid chromatography mass spectrometry more efficient? So researchers have been trying various solutions to decrease the cost of mass spec analysis or to maximize what they're achieving from this resource, and also to harness the sensitivity and selectivity, which is the greatest advantage of the mass spectrometer for better data, for better output. So many of the solutions have revolved around making the liquid chromatography step more efficient. So things like UHPLC, where the chromatographic system is micronized, resulting in a faster cycle time. This often also manifests in less solvent use and less waste. Multiplexing LC systems, so this is staggering your injection into the mass spectrometer and using multiple LC systems, because a liquid chromatography system is a much less expenditure than a mass spec. This has been another solution. And then increasing the flow rates to kind of speed up the chromatography step in the process has also been a strategy that has been used. But what I want to talk about today is another strategy, which is using, rather than liquid chromatography, using SPE, or solid phase extraction, with a combination of excellent engineering, and that is the RapidFire mass spectrometry system I'm going to talk about today. So RapidFire is an extremely fast purification system composed of integrated microscale extraction. Thus, RapidFire is replacing the liquid chromatography portion of the LC-MS system. So it's without a full gradient, which chromatography has, but this results in samples being processed by the RapidFire system in as little as 6 to 10 seconds each, which is nearly 10 times faster than traditional HPLC. In fact, the RapidFire system is also compatible with a lot of different matrices. So many people think separation is absolutely required in chromatography, but we've shown that in many occasions a cleanup by solid phase extraction so that the mass spec can handle the sample is all that's necessary. So we've been able, with RapidFire, to use input types like microsomal preparation, cell culture supernatant, tissue extract, plasma, whole blood, urine, and like I said, a variety of different matrices. So how does solid phase extraction work? So that's what this slide demonstrates. So basically, with an aqueous wash, analytes of interest bind onto the SPE cartridge, while large particles and other components of the assay continue on to waste. RapidFire cartridges here are available in a wide variety of chemistries to suit the binding of whatever particular analyte you're looking at. These analytes are then eluted with an organic wash, so they're reverse eluted off of the cartridge right into the mass spec where the analytes of interest are detected. So let's take a closer look at the RapidFire system. So this graphic shows the system working with, this here is a 6410 triple quadrupole mass spectrometer. Our system works, the RapidFire system integrates with any mass spectrometer, any Agilent mass spectrometer, be it a triple quad, a TOF, or a Q-TOF, like Yuqin just talked about earlier. The RapidFire system It's composed of a microfluidic cleanup system, which has proprietary valves that are made of a diamond-like coating. So what this enables is these valves to maintain stability over millions of switching cycles. So these, you can imagine at 6 to 10 seconds per sample running plates all the time, these valves are constantly switching, and they're much more robust than traditional valves. A plate handler is necessary, obviously, to feed the system, and the system can accommodate 96 or 384 well plates. All the connection is via peak tubing directly to the source, so directly to the source of the mass spectrometer, which you see the connection here. So very similar to any LC system. The system also, it's kind of hard to see in this picture, but incorporates a cartridge changer which handles up to 6 cartridges at the same time, or 6 cartridges that can be changed when the pressure builds up on one cartridge, or however one would like to set it, and this enables overnight runs. So now what I want to show you is how we feel that RapidFire has really evolved working on the mass spectrometer. So this is an HPLC run. So here we're looking at a 3-minute run. Here's your peak of your sample of interest, which in here is the internal standard. And then here is hydroxymidazolam, which is the product that we're looking for in this reaction. And here you see this is a runtime of about 3 minutes per well. So if we were going to go through an entire 96-well plate, it would take us approximately 5 hours. So in the same time frame with the RapidFire system, where we're going much faster at 6 to 10 seconds per sample, you can see it would say the same internal standard and the same buildup of product over time, but at 10 times the speed. So basically, this screen capture here shows again the same peaks, but this is the entire 96-well plate. So 96 samples, but this entire plate at 7.5 seconds per sample took less than 12 minutes for this 96-well plate. So this is a great increase in speed using the RapidFire system. So now I want to show you a very brief video, okay, which doesn't seem to want to start. Hold on, I need to click it here. Okay, well, it was working fine before I went on to the webinar, so it must have something to do with being on the webinar. But this is a close-up, basically, of our valving system right here, and this is the plate handler. So what happens is the plate handler picks up a plate, brings it over onto the surface of the instrument here, onto this stage, which comes down under our sipper tube or needle, which will grab a sample using a constant vacuum. The sample will go up into the cartridge, bind onto the cartridge, and then the flow will move on to waste. Valves will change. We will wash with the organic solution and back elute that sample onto our mass spectrometer. So now that I've told you a little bit about how the RapidFire system works and some of the background of the system, I wanted to focus a little bit on the markets and the applications of the RapidFire system. So lead discovery is one of our very important applications. So lead discovery encompasses primary screening, secondary screening, SAR, enzymatic, kinetic parameters, and IC50s. RapidFire has also been used very successfully for in vitro ADME assay analysis, things like CYP450 inhibition, metabolic stability, permeability, and PGP inhibition. Finally, clinical research and forensic toxicology, applications such as drugs of abuse, pain management, some of the forensic tox analysis that is done in a variety of those types of molecules. So now I want to dig a little bit deeper into the lead discovery application of RapidFire technology. So traditionally, in the lead discovery market, when looking for new drugs, the traditional method of choice is a plate reader-based assay with optical or radioactive readout for its ease of use and analysis speed. However, as we mentioned earlier when we were talking about the benefits of mass spectrometry, sometimes it becomes difficult when we have to use optical, radioactive, fluorescence in order to get excellent readouts. So, but again, the balance is always the speed. But when we use the RapidFire system, we're able to get very close to that speed of a plate reader, but again, taking advantage of this excellent tool, the mass spectrometer. So where is RapidFire really excellent for lead discovery, where we've actually enabled more targets and targets that were very difficult to do using optical assays? One is for native analyte detection. So here we're looking at surrogate substrates that can introduce confounding factors, affect enzyme kinetics, and produce data artifacts. And in fact, we've seen this in several cases with our collaborators. When they were using optical assays, they were not getting accurate results. So here using native analytes, so detecting the native analyte as opposed to using a surrogate, has really helped to achieve success for some of our collaborators. Then there's the second example where we're replacing an intractable assay. So there's a lot of assays, for example, that present challenges to the laboratory workflow. They might be resource intensive, cost prohibitive, and may present regulatory issues. So maybe there's radioactivity and the laboratory doesn't want to be using very much radioactivity. So we call these intractable assays, assays that would be very challenging. Well, RapidFire is able to get around a lot of these issues. And then the third, another very important reason that some of our collaborators go to use RapidFire is RapidFire's ability to enable various target classes. So we're talking about things like multiple modification events on a single substrate, things that are very impossible to track by many common optical and radioactive methodologies. RapidFire has proven very successful. So I'm going to show you a couple examples of those as well. So what we're looking at here is a reaction, and this is RapidFire, an example of RapidFire enabling epigenetic target classes. So the field of epigenetics focuses on investigation of enzymes that alter gene expression through modification of their target substrates, frequently by adding or removing acetyl groups. So historically, studying the activity of such enzymes in a high-throughput manner has been very challenging due to relatively small molecular alterations that we're looking at. So here we're looking at the addition of acetyl group. Also, the possibility of sequential modifications, which can lead to multiple analytes of interest. So this example here is the enzyme P300 histone acetyltransferase, which catalyzes the reaction of adding an acetyl group onto a protein using acetyl-CoA. So what happens, for example, in this case, is if we're looking at, this is just a small portion of the P53 protein, so just the peptide. But if we're looking at this peptide, there are 4 potential preferred sites where the enzyme P300 would choose to acetylate P53. So you can imagine if we're using traditional methods, we're looking at binding of a fluorophore and then a reaction that's taking place where an acetylation occurs. Even if we're using an antibody to an acetylated site, it would be very difficult for us to detect acetylation at a single site, for example, versus acetylation at 2, all 3, or maybe 4 of these sites, and of course any combination therein. However, with RapidFire, this is proved very easy to do. So here's some data demonstrating this. So here, this is RapidFire data with a triple quadrupole mass spectrometer. So you can see what we're looking at is sequential acetylation over time. So as we combine this P53 peptide with P300, the blue line is 0 acetylation. So at time 0, the peptide is not acetylated by P300. And over time, it decreases, the blue line, as acetylation is taking place. So red is 1 acetylation, green is 2 acetylation, 3 acetylation sites on the peptide, and eventually the yellow is 4 acetylation sites. So you can really see the sequential change over time. Now imagine trying to do this using an optical assay or an optical readout. So this is one example of where RapidFire has really enabled detecting a reaction that was very difficult to detect by other means, or impossible. In addition, we've also analyzed the whole protein, whole histone protein. So before we were looking at a peptide, now we're looking at a whole histone protein. In this case, we're using time-of-flight mass spectrometer, where we can look at these larger whole proteins. And so what we've done here is use the RapidFire TOF to illustrate a range of options for measuring histone acetyltransferase activity using high-throughput mass spectrometry. So this is the inhibition of P300 by garsenol, which is an inhibitor. So we can see the concentration change in garsenol using whole histones as a substrate. And this is deconvoluted RapidFire TOF data for 18 reactions. And here we're looking at IC50 for the 6 concentrations in triplicate, and what's indicated is the increasing acetylation with decreasing inhibitor concentrations. You can see as the inhibitor concentration increase, we have a decrease in the amount of activity of the enzyme. And it's very nice to be able to compare 0 acetylation, 1 acetylated histone, twice acetylation, depending on the different amount of inhibitor, and to really see this when we're looking at the whole protein molecule, to really show that we're seeing on the whole protein what we were seeing on the peptide. So that's just one example, and I just wanted to let you know that RapidFire has been used to evaluate a wide range of drug discovery targets for a variety of different therapeutic areas, which are listed here. These are just a sample. These are some of the targets in therapeutic areas which have been published by one of our team or by some of our collaborators. Now I want to switch to a different application area. This is the in vitro ADME assay analysis. So we also have a lot of collaborators who are working on in vitro ADME using RapidFire. And these are for analysis of assays such as CYP450 inhibition, metabolic stability, permeability, and plasma protein binding. Traditional in vitro ADME assays are analyzed using liquid chromatography mass spectrometry, and RapidFire thus can process the analysis approximately 10 times faster. So we're talking about lead discovery being a plate reader-based traditional screening type of assay, and RapidFire being able to accomplish working in a label-free environment and really taking advantage of the mass spectrometry. RapidFire on the in vitro ADME assay side has been able to take what's usually done by liquid chromatography mass spectrometry and speed that up, so enabling the decrease of bottlenecks for some of these assays which are already using chromatography and mass spectrometry. So of course, one of the things that we always try to do is we need to compare using RapidFire to the traditional methods. So here we're looking at a RapidFire metabolic stability analysis. So here, and we're looking at comparing using the RapidFire in a time-of-flight system versus LC with triple quad mass spec. And we're looking at a diverse set of 39 drug-like compounds. So these are compounds with lots of different characteristics and properties. So we want to make sure that the full range is able to be detected similarly by RapidFire and using an LC system. You can see what's important here is the bottom, is that for a RapidFire TOF analysis is occurring in 9.5 seconds per sample, whereas LC triple quad takes 2.2 minutes per sample. So there's quite an increase in speed using the RapidFire system. Now here's the data. So we're looking at results for 2 of these compounds. So of the 39 compounds, here's just 2 representatives, nicardipine and midazolam. So as you can see, RapidFire mass spec in orange compared to LCMS in blue show very similar half-lives in this metabolic stability assay for both of the 2 compounds we looked at. So excellent correlation between the 2 systems. Now, I told you that what's really important for RapidFire and in vitro ADME is the ability to process faster or reduce bottlenecks in sample analysis. So one of our collaborators had presented this at ASMS a year or so ago, and this is a typical workflow in this investigator's laboratory working on in vitro ADME analysis. They would receive their samples from medicinal chemists on Monday. They do tuning of the analyte, sample analysis by LCMS, and deliver the data about 38.5 hours later, or after approximately a week of work. They would deliver the data back to the medicinal chemist, and they could be ready to go on to the next round of developing this target molecule. So the lab incorporated the RapidFire 300 system. So this is the RapidFire system that works with the triple quadrupole mass spectrometer. So what they were able to do is decrease this 24-hour LCMS time down to 2-hour time frame. So now it's taking less than half a week for the data to be sent back to the medicinal chemist. Well, then this group then incorporated the TOF system, so the RapidFire 360, which integrates with the TOF, or the time-of-flight system. So this enables workflow where a generic method can be used such that analyte tuning is not necessary for each particular compound. So in this case, this laboratory now receives their samples on Monday morning. By Monday afternoon, they're able to deliver the data back to the medicinal chemist for the next round. So you can imagine in the drug discovery process where it's laborious and very expensive, costing millions to billions of dollars, anything that shortens this development time is really going to save time and money to the drug discovery company. So this is a very good example of where RapidFire has been able to reduce the bottlenecks and reduce the analysis time from almost a week to less than a day. So of course, something that's very important when we're talking about RapidFire, and I told you what some of the traditional methods were, whether they're optical assays or LCMS assays, it's extremely important that we're able to have great correlation analysis with these traditional methods. So here's an example of doing an IC50 using RapidFire versus HTRF. So this is a market-leading fluorescence assay, and you can see excellent correlation. Another example here, we're looking at CYP inhibition. So here, RapidFire MS, this is an in vitro ADME assay, or LCMS. You can see, again, a nice correlation. This is looking at an anticonvulsant compound. So here we're demonstrating that even in matrices such as human serum, we're able to see a nice correlation in results between RapidFire and LCMS. And then this is PAMPA. So PAMPA analysis is another type of in vitro ADME assay. And we're demonstrating here that whether you use a triple quad method for analysis or the TOF method for analysis, so two different types of mass spectrometers, we're able to get good correlation using the RapidFire system. So when we think of how we can work closely with our collaborators, we get to two main pain points, basically, that our researchers are having with their workflows. The first one is the problem that they have too many samples. We have sample bottlenecks. But what RapidFire is able to do is to increase throughput, enable them to go through and process these samples faster. I also hear collaborators and researchers telling us, well, they have the need to report results faster. So, okay, maybe they don't have hundreds of thousands of samples, but they need a really quick turnaround time because they need to get these results out of their laboratory as quickly as possible. So that's another thing that RapidFire has been able to assist our collaborators with, is increasing this turnaround time. So just as a brief conclusion, RapidFire does have the ability to resolve bottlenecks. It improves data quality, so high throughput. Researchers are able to do more concentrations or more time points within the same amount of time. And it also avoids surrogates, substrates, indirect, and coupled assays. So like I mentioned, optical, fluorescent, or radioactive readouts. 10 times faster, we're really able to increase the productivity of the labs that we work with. Assay development is very quick as well. It's something I didn't have time to really go over too much in this presentation. And I showed you that RapidFire has a strong correlation with traditional technology. So we're not really replacing there's no compromise to your data if we're using RapidFire as opposed to traditional technology. Other advantages of RapidFire are its lower operational costs. So there's a lot of solvent savings. Cartridges have a lot of have quite a lifespan. So a lot of samples can be produced through one cartridge. Minimal reagent disposal costs, so there's no radioactivity, no kits or reagents. And we also fit existing workflows. So the RapidFire is designed to operate very similar to a plate reader so that our collaborators are looking at a system similar to what they're used to. So at 6 to 10 seconds, it has a very high throughput, a very low cost, and ease of operation, being compatible with standard libs and having a similar structure to current laboratory workflow. Because we're talking about increasing the efficiency of your mass spectrometry system, just a reminder that RapidFire works with triple quadrupole instruments, TOF instruments, and Q-TOF instruments, and we have integrated workflows with RapidFire and the Agilent MassHunter software that Yuqin was talking about, giving our collaborators the ability to deliver data in hours rather than days. So I just wanted to say thank you very much, and we'll turn it over for any questions.
Thank you, Jennifer. If you have any questions for Jennifer, please send it in. You can type it into your question box. I also wanted to remind everyone that today's webinar will be available for viewing on Lab Manager's website for 6 months. The link to the archived webinar will be emailed to all of you sometime next week. The email addresses of our speakers will be shown to you shortly. So feel free to reach out to them for a copy of their presentation, or if you have any additional questions based on their talks. So let's begin by talking about sample prep. I know that both of you did address sample prep a little bit in your talks, but can you elaborate a little bit about the key issues around sample prep, sample carryover, cleanup, and how attendees can minimize some of those problems? But yeah, go ahead.
So this is Jen. It's very different. Yuqin talked about liquid chromatography, and what I was talking about is SPE. And they're both sample preparation methods. Chromatography, obviously, there's much more of a separation between of the sample, and SPE has much more. It's much quicker. And for desalting purposes, basically, there's no gradient involved, at least what we're talking about here with RapidFire. It's basically a quick binding onto the cartridge chemistry and then elution off. Mm-hmm. So the cleanup is very different between the two. Yuqin might want to comment a little bit more from the chromatography side.
Yeah, for the work I'm discussing today, actually, sample preparation is very generic. Just use the acetonitrile protein precipitation. That's all. For this in vitro sample, actually, we just use acetonitrile to stop the reaction and then further dilute in the solvent, aqueous solution, and then just directly inject to the LC mass spec. So I don't really do any sample. This sample preparation just very generic method. For the plus sample, again, I just use the acetonitrile, to extract the analyte. Just protein precipitation. I don't using any rigorous sample cleanup method.
Okay.
And so you had also mentioned carryover in that section, and for the RapidFire system, between every sample, there is an aqueous and an organic wash of the sipper tube that basically eliminates any carryover, as well as the fact that the system's composed of peak, which is also built in to help to eliminate any carryover issues.
Right. And how often do these cartridges have to be replaced, and how do you know when they need replacement?
So it depends a lot upon what your analysis is. So what type of samples you're putting through the system. But in general, 2,000 to 3,000 injections per cartridge. And I briefly mentioned the fact that we have a cartridge changer which holds 6 cartridges. These can be of the same chemistry or of different chemistries. And we have an optical SIP sensor that also measures the liquid fluid flow through the system. And we are able to detect any pressure buildup in the cartridge. So the system can be set however you would like to. You can say, please change cartridges every 3,000 injections. Please change cartridges when the pressure builds up to this particular point. Or if the pressure really builds up, it will change automatically.
Got it. Okay, thank you. And how do the two systems, like if you have a LC front end compared to the SPE front end, how do they compare in terms of cost?
In terms of cost, so yeah, the RapidFire system is about 3 or 4 times more expensive than an LC system. So it is. So the RapidFire system is about the price of another mass spec. So it definitely has a much more significant cost. But also that higher throughput. So 10 times faster than one mass spec costs, than one LC system costs about, the amount of 3 to 4 mass LC systems.
Okay, got it. And now, just to clarify, the RapidFire can only be used with other Agilent mass specs, correct? They can't be tied in with other mass specs from other companies.
We do have an integration with some additional mass spectrometers, namely SciEx triple quadrupole mass spectrometers. We can also interact with those. It's a simple contact closure for anything else. When it comes to accurate mass or TOF instrumentation, we only integrate with the Agilent TOF mass specs. But you can also see the advantage of using the Agilent mass spec, the triple quad or TOF or Q-TOF with the RapidFire system because of the integration of the software. It makes for a much more seamless integration, but yes, it is possible on the triple quad side to use a different flavor of mass spec.
Okay. And, this question, I guess, is again for both of you. What types of customer training is provided?
What type of training?
Customer training. I mean, do you train people to use these instruments? Is there on-site training, or do they, do people have to come to your facility to get trained?
Okay, I can start for the RapidFire side and hand it over to Yuqin if you'd like. So for RapidFire training, upon installation, someone comes and trains the users on site. We also offer additional training programs in method development, assay development for different compound types that can be tailored to the specific needs of the customer. We also have demo labs as well where we can bring people in and show them the system.
Okay, go ahead. Yeah.
Yes, same thing for the LCMS. We actually have very great support group, and we also have demo lab and technical support through TC, and also we have the training center, provide a training course for hands-on training, theory training, everything customer need support, Agilent always give. That's not a problem.
And can people send you samples so that they can compare, with what they are getting with their current mass specs right now and see how it can be improved, or, what resolution and sensitivity your methods and your products offer?
Yes, definitely. We have the product specialist and the product account manager. They will arrange this, sample demonstration in our COE lab. Demonstrator lab. So this definitely, if some people are interested to do that, you're welcome, to contact to us.
Okay.
And it's the same situation with RapidFire as well.
Okay. All right. Yeah, I was just about to ask that. All right. Well, we are unfortunately, over a little bit over, so I have to end our webinar. But thank you so much for putting in the time and effort to get your presentations ready and make it so very informative and relevant to the users. So again, on behalf of Lab Manager, I would definitely like to thank Agilent Technologies for both their participation and support. As you can see on your screen, you have the email addresses of the speakers, and if you need a copy of their slides or if you have additional questions, definitely reach out to them. The webinar will be archived on Lab Manager's website, so do visit us online to review this webinar, past webinars, or to get information about upcoming webinar topics. I hope you can take the time to join us again for our webinar later this week, which is going to be on February 23rd. And we will be discussing innovations in ultra-low freezer technology then. So thank you all once again for your time and attention. Until next time, goodbye and good luck.